And one of the problems, particularly in the neurointensive care world, was that the doctors and nurses don't always see the patients during the recovery phase. I developed BIOS to get long-term outcome data on acute illness. As time went on, we realized that independent of your area of specialty, all translational physicians needed turnkey operations that understood all of the learnings that have gone on, predominantly in the pharmaceutical and device industries, but also at universities. And we've embraced learnings from all three of those areas to develop BIOS into what it is today, which is actually the Johns Hopkins clinical trial unit. We currently operate 20 active trials where we do the operations of the trials, with about 30% of our trials being brain-centric and the rest agnostic to specialties, everything from neonates to aging and dementia trials. What we're doing now, we were lucky enough to devote ourselves fully to one task, which is doing trials better and learning from the mistakes that we knowingly and unknowingly made. And if you look at each trial as an instance, you can criticize yourself and your team and the environment and try to do things differently the next time. What is the way forward? It's trial science, which is something we've been doing for the last 15 years. And that involves using the metadata that comes out of trial operations and looking just like you would in any business. It's a manufacturing process, and how do you make it better? Most of what we do, we've learned by doing pharma trials and device trials and applying those lessons to academic trials. And I think there are some things that pharma and the device world can learn from the academic world, particularly in the area of explanatory trials. Trials that build a background of information around which you can reorient a neutral phase 3 into a positive phase 3. Many goal-directed enterprises don't want to invest in a broader set of explanatory data gathering. But I think in the world of proteomics and genomics and rare diseases, we have to admit we don't have the phenotype right on the first, second, or third pass. And there are multiple examples throughout the COVID pandemic of where we got the timing and the dose wrong as well. I think there's a strong role for what academics like to do, what has been called sometimes small shitty trials that gather too much data. There is a role there, particularly in the world that we're entering.